Hello dear students, today we are going to discuss the topic phloem loading. The main objectives of the present topic are: number 1, to understand the concept of phloem loading; number 2, to differentiate between apoplastic and symplastic mechanisms of phloem loading; number 3, to get familiar with different types of sympotors involved in apoplastic phloem loading. Number 4, to understand the symplastic phloem loading with the help of polymer trapping model. And number 5, to differentiate temperate and tropical plants on the basis of their phloem loading mechanisms. First of all, a brief introduction about phloem loading. The transport of photoassimilates from the mesophyll chloroplast to the sieve element companion cell complex of leaves is known as phloem loading. It includes various steps like the photoassimilate synthesizer during the day in the form of triose phosphates are transported from the chloroplast to the cytosol. In the cytosol, these triose phosphates are converted to sucrose. However, during the night, the carbon from stored starch exits the chloroplast in the form of glucose. This glucose is then converted to sucrose in the cytoplasm. Number 2. This movement from mesophyll to nearest sieve element occurs in the smallest veins of the leaves. During this transport, sucrose traverses a distance of only 2 or 3 cells. Number 3. During phloem loading, the sieve elements and companion cells acquire sugars from mesophyll cells. It has been found in most of the species that the sugars become concentrated more in the sieve elements and companion cells than in mesophyll cells. Sieve element and companion cell act as a single functional unit called sieve element companion cell complex during the loading of sugars in the source tissues. Inside the sieve elements, sucrose and other solutes are translocated to the sites where food is needed. This process of transferring food to sinks is known as export. It's a long distance transport that takes place through the vascular system. The process of loading photosynthetes at the source and its unloading at the sink generate the driving force for this long distance transport. Phloem loading is thus considered as one of the basic processes on which agricultural produce depends. Understanding these mechanisms can help us enhance crop productivity. One of these mechanisms involves how we can increase accumulation of photosynthate in edible sink tissues such as cereal grains. Now let's see how photosynthates are loaded. Phloem loading can occur either through apoplast or the symplast. The movement of sugars in source leaves to the veins of the same leaf takes place through different routes. These sugars can move entirely through the symplast, that is cytoplasm, using plasmodesmata, or they may traverse both symplast and apoplast. In the later case, sugars enter the apoplast at some points before reaching the sieve elements. The sugars present in the apoplast are actively loaded into the sieve elements and companion cells by using energy. There is a specific transporter for this purpose, which is located in the plasma membranes of these cells. Usually, the apoplastic and symplastic routes are used in different species. It means both these pathways are rarely present in the same species. Now let's move towards the apoplastic phloem loading. The movement of photosynthate from the mesophyll chloroplasts to the sieve element companion cell complex of matured leaves via cell wall and intercellular spaces is known as apoplastic phloem loading. Plants having apoplastic phloem loading have ordinary companion cells or transfer cells in the minor veins. They are able to transport sucrose only. There are three basic predictions regarding apoplastic phloem loading, which include: number 1, the sugars which are to be transported should be present in the apoplast, that is, the space outside the cell membrane. Number 2, if sugars are supplied exogenously to the apoplast, they should accumulate in the sieve elements and companion cells. Number 3, export of photosynthate from the leaf to the sink should stop if uptake of the sugars from apoplast is inhibited. Let's see now role of ATP in apoplastic pathway. Accumulation of sucrose in the sieve element companion cell complex requires metabolic energy in the form of ATP as it moves against its chemical potential gradient. It has been observed that treatment of source tissues with respiratory inhibitors decreases ATP concentration. This decreases the pace of sugar loading in the sieve tubes at the source. Unlike sucrose, some other metabolites like organic acids and hormones enter the sieve elements passively. The difference in solute concentration between sieve element companion cells and mesophyll cells in the source leaves is due to phloem loading. This difference in solute concentration is demonstrated mostly by using osmotic potential measurements of sieve elements and mesophyll cells. For example, in sugar beet, the osmotic potential of the mesophyll is about minus 1.3 megapascals. Sieve elements and companion cells, on the other hand, were found to have osmotic potential of around minus 3 megapascals. The accumulated sugar in sieve elements has been found to be sucrose because it is the main transport sugar in this species. Now, the role of sucrose proton symporter in sieve element loading. A sucrose H-positive symporter mediate the transport of sucrose from the apoplast into the sieve element companion cell complex. Sieve element loading via apoplast uses the energy generated by the proton pump. Therefore, sugar loading through sucrose H-positive symporter is a secondary active process. The movement of protons through the symporter is coupled to the movement of sucrose inside the cell. It has been observed that high pH means low H-positive concentration in the apoplast reduces sugar uptake in plants. This is due to a reduction in magnitude of H-positive ion concentration gradient. In other words, the driving force for sucrose loading gets reduced at high pH. Molecular studies also support the operation of a sucrose H-positive symporter in sieve element loading. proton pumping ATPases are localized in the plasma membranes of companion cells of Arabidopsis. These ATPases were also found on the plasma membranes of transfer cells of broad bean. Most importantly, these pumps are localized more in the plasma membrane enfoldings that face the bundle sheath and phloem parenchyma cells. The function of these H-positive ATPases is to energize the transport of photosynthate from the apoplast to the sieve elements. In Arabidopsis, the distribution of the H-positive ATPases appears to be correlated with the distribution of a sucrose H-positive symporter called SUC2. This sucrose H-positive symporter has also been found in companion cells of some other plant species like Plantigo major. H-positive ATPases and sucrose H-positive symporters are also co-localized in the plasma membranes of sieve elements. But this arrangement has not been found in majority of the species. Among the various sucrose H-positive symporters, SUC2 has been cloned and localized in the phloem. The carriers which are found only on the membranes of sieve elements include SUT1, SUT2 and SUT4. On the other hand, SUC2 has been found on the plasma membranes of companion cells. The mRNA for the SUT1 is formed in the companion cells and the protein synthesized is then localized on the plasma membrane of the sieve elements. This supports the non-nucleated nature of mature sieve elements and their dependency on the companion cells for protein synthesis. It has been shown that most of the symporters are present in the source, path and sink tissues. SUT1, characterized as a high affinity, low capacity transporter found in the minor veins of source tissues, appears to be important in phloem loading. The sugars which are lost in transit are also retrieved by SUT1. Another important symporter, SUT4, complements the function of SUT1. SUT2 has the function of sensing sucrose and it is more highly expressed in sink and part tissues than in source leaves. This symporter has many structural similarities with the sugar sensors. SUC2 functions in the uptake of sucrose into the companion cells. Now let's have a look how sucrose loading is regulated. There are many mechanisms which explain the regulation of sucrose loading from the apoplast to the sieve elements. Some of the most important regulatory factors in sucrose loading by the sucrose H-positive symporter are: number 1, the solute potential or the turgor pressure of the sieve elements. Decrease in sieve element turgor increases sugar loading as a compensatory measure. Number 2, sucrose concentration in the apoplast. High sucrose concentrations in the apoplast increase the sucrose loading. Number 3, number of symporter protein molecules. The levels of SUT1 transporter mRNA and protein have been shown to be lower in darkness than during light periods. This suggests the role of SUT1's transporter molecules in regulating the process of phloem loading. Number 4. Mineral ion availability. Availability of potassium ions has also been implicated to affect the efflux of sucrose from mesophyll cells into the apoplast. This suggests that a better nutrient supply enhances the transport of sugars to sink tissues and increases their storage and growth. Now let's move towards the symplastic phloem loading. The movement of photosynthate from the mesophyll chloroplasts to the sieve element companion cell complex of mature leaves via cytoplasm is known as symplastic phloem loading. Symplastic pathway of phloem loading occurs in species which transport raffinose and sticose in the phloem. These transport sucrose also, but not as much as raffinose and sticose. These species have intermediary cells in the minor veins. Common coleus, squash and melon are the examples of plant species which load sugars symplastically. Symplastic loading of sugars is possible only in presence of open plasmodesmata between different cells in source tissues. Numerous plasmodesmata are found in these species between the sieve element companion cell complex and the surrounding cells. Now let's see the mechanism of symplastic loading in source leaves. Differences in the composition of sugars in the sieve elements and the surrounding tissues of the source leaves has been experimentally observed. This shows that certain sugars are selected for transport from the sources to the sink tissues. Use of sympoters in apoplastic phloem loading gives a clear indication of specificity. This is due to the fact that all sympoters are specific to the specific sugar molecules. In case of symplastic loading, however, movement of sugars takes place due to diffusion from mesophyll to the sieve elements. This transport of sugars takes place through plasmodesmata. However, simple diffusion is not enough to explain symplastic phloem loading. This is due to the fact that during symplastic loading, only some selected sugars are loaded into the sieve elements. Polymer trapping model. This model has been proposed to explain as to how specific sugars are transported in diffusion-based symplastic loading. According to this model, sucrose synthesized in the mesophyll diffuses from the bundle sheath cells into the intermediary cells symplastically. These cells are connected by abundant plasmodesmata. Inside the intermediary cells, raffinose and sticose, which are the polymers made of 3 and 4 hexose sugars respectively, are synthesized. These sugars are formed from sucrose and galactose. Due to particular anatomy of the tissues and relatively large size, raffinose and sticose, the polymers cannot diffuse back into the bundle sheath cells. However, they can easily diffuse into the sieve element because of wider plasmodesmata between companion cells and the sieve elements. The utilization of sucrose in the intermediary cells in polymerization reaction and its continuous synthesis in the mesophyll cells maintains a concentration gradient that is essential for symplastic loading. Different predictions of polymer trapping model are: number 1, concentration of sucrose must be more in mesophyll cells than in the intermediary cells of the source tissues. Number 2, the enzymes for raffinose and stacose synthesis must be present in the intermediary cells only. Number 3, plasmodesmata between bundle sheath cells and the intermediary cells should exclude molecules larger than sucrose. Various experimental observations from symplastic phloem loading studies are: most of the studies are in support of polymer trapping model. Number 1, enzymes for the synthesis of stacose from sucrose and galactose have been found in intermediary cells. Number 2, in melon, raffinose and stacose were found in higher concentrations in intermediary cells compared to mesophyll cells. Number 3, higher concentration of sucrose has been found in mesophyll cells as compared to intermediary cells. Finally, let's see the correlation of phloem loading with plant family and with climate. Now, it's clear that the operation of apoplastic and symplastic phloem loading pathways are correlated with the following features: type of transport sugar, the type of companion cell in the minor veins, and the number of plasmodesmata connecting the sieve elements and companion cells to the surrounding photosynthetic cells. Species which have apoplastic mode of phloem loading translocate sucrose almost exclusively. They also have either ordinary companion cells or transfer cells in the minor veins. Finally, they are known to have a fifth plasmodesmatal connections between the sieve element, companion cell complex and the surrounding cells. On the other hand, species which have symplastic mode of phloem loading translocate oligosaccharides such as raffinose in addition to sucrose. These plants have intermediary type companion cells in the minor veins. In addition to this, they possess abundant plasmodesmatal connections between the sieve element, companion cell complex and the surrounding cells. Plants which have abundant plasmodesmata between the phloem and surrounding cells are often trees, shrubs or vines. However, only few plasmodesmata occur at this interface in herbaceous plants. Plants with abundant plasmodesmata between the phloem and surrounding cells are prevalent in tropical and subtropical regions. The plants with few plasmodesmata at this interface are found predominantly in temperate climatic zones. There are, however, some exceptions to these arbitrary generalizations. For example, some species with apoplastic loading have more plasmodesmata linking their companion cells to surrounding cells. According to apoplastic theory, they should be having only few plasmodesmata between companion cells and surrounding cells. Most of these species are known to have more than one type of companion cells in their minor veins. Common Coleus, for instance, has both intermediary cells and ordinary companion cells in its minor leaf veins. Symplastic and apoplastic pathways are also believed to coexist in some species. This coexistence may occur simultaneously or at different times. It is believed that future studies may help understand loading pathways or combinations of pathways more accurately. The evolution of different loading types and how these types adapt species to their environment is an important future dimension in phloem loading studies. This was all about today's lecture. Thanks for being patient.